So we picked bananas and apples to represent our chromosomes and centromeres. We chose this approach because food is an easy thing to get and it's just like a good visual representation, especially for people who like to learn visually. This is a really good approach because you're actually able to look at something and move things, and it's also good for people who like to do stuff with their hands, and it's easier for them to remember it that way. The bananas represent the chromatids of the chromosomes, the apples represent the centromeres, the Skittles represent the centrosomes, and the spaghetti represents the microtubules.
In interphase of meiosis 1, we begin with unduplicated chromosomes, and at the end of interphase, they duplicate in DNA synthesis, which is represented like this, and then they become duplicated chromosomes. During prophase 1, they become homologous pairs, and also during this phase, crossing over occurs, which we will not be showing, and the pairs are formed in random assortment, so you can't necessarily know what it's going to become. During metaphase 1, the homologous pairs line up in the center, and then the microtubules form, the mitotic spindle forms, and they attach to the kinetochores of the chromosomes. During anaphase 1, the homologous pairs separate and go to opposite poles. During telophase 1, the nuclear envelope forms around each of these, and they become two separate diploid cells.
Between meiosis 1 and meiosis 2, there is no interphase, and in prophase 2, the nuclear envelope breaks down and the chromosomes condense. In metaphase 2, the mitotic spindles form and they connect to the kinetochores, and the chromosomes align in the middle. In anaphase 2, the sister chromatids separate and they move to opposite poles. In telophase 2, the nuclear envelope reforms and we now have 4 haploid cells.
So in the F1 progeny, we cross the homozygous white fur with a heterozygous black fur. If we look at the Punnett square, we get 50% black fur and 50% white fur. The black fur is dominant and the white fur is recessive. So in the F2 progeny, we are crossing the homozygous white fur, and in the end we get 100% recessive white fur.